Hi, I'm Liliana Vasquez, and today I will be talking about DNA replication. DNA replication is one of the basic processes that goes on in a cell. The replication is the process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. The double helix strand is used as a template for the new forming DNA. Each time a cell divides, the two resulting daughter cells must contain exactly the same genetic information or DNA as the parent cells. Telomeres also help with that by playing a good role during the process and protecting the important genes from being deleted as cells divide and as DNA strands shorten while replicating. To achieve the proper DNA replication, several steps need to occur. Step 1, the separating of the strand. This is the untwisting of the double helix strand to turn into a single molecular strands. It happens in the specific area of a chromosome known as the origins. The DNA strands are being separated by breaking the hydrogen bonds between the nucleotide base pairs such as adenine and thymine, guanine and cysteine. Those are the typical ones. It then goes to step 2, which is replication fork formation. This is the splitting of the double helix that represents a Y-shaped formation known as the replication fork. So basically, after the helix is split, you're able to see a Y-shaped figure. Each side of the Y shows an elongated line that needs an identical half to match up with it so it can form a new pair of strands. One of the separated strands is called the leading strand, which is constantly utilized for DNA synthesis, while the lagging strand is represented for the complementary strands synthesis. Then comes step 3. Step 3 is the binding of base for each strand. Both the leading strand and the lagging strand already have base patterns from the start, and it serves as the template for each corresponding strand. For each strand, the bases match with the free-floating nucleotides present inside the cell following the nucleotides base pairing rules to establish the hydrogen bonds between a separated strand and a new matching strand. So when that happens, nucleotides that are just floating around in there match up to whichever patterns it can, that would correspond with it. It is then made by DNA polymerase, which is an enzyme that matches and zips both strands together. The last step is the termination of the replication process. This occurs as soon as the DNA polymerase arrived at the edge of the strand and there is no more possible replication to occur. But before the process is completed, it goes through the process to repair and correct errors such as mismatching of nucleotides. After this step, the DNA replication is complete.